How's it going, everyone? It's Chow here, and today we are going to talk a little bit about light-dependent reactions, so let's get started. Okay, so light-dependent versus light-independent reactions. A good question that always gets asked. Both of these occur in the chloroplast, which is also where photosynthesis takes place. So light-dependent reactions are going to be on the thylakoid membranes, and the byproducts are going to be oxygen. In the light-independent reaction, it's going to occur in the stroma, and the overall byproducts are going to be sugars. So the thylakoids here are basically these little individual discs, and each stack is called a grana. And on each membrane, there is a series of proteins which can cause the reaction to actually occur. So we have pigments over here. And pigments are basically molecules that are able to absorb specific wavelengths in the visible range. And then it's basically others are going to be scattered or going to be transmitted. So it only absorbs specific wavelengths. A very well-known molecule that's a pigment is called chlorophyll, and it absorbs blue and red light and reflects green light, which should make a lot of sense. So when we look at the light reactions themselves, we're really converting light energy into chemical energy, and this is sometimes called the light cycle, and it happens again in the chloroplasts. The chloroplasts are very similar to mitochondria, but also slightly different. They're quite similar in that they have a double membrane, and what's really fascinating about chloroplasts is that some people think that they also used to be free-living organisms that through the endosymbiotic theory became incorporated into the larger cell. So we have proteins in the thylakoid membrane that are embedded that absorb light, and they basically allow for this reaction to occur over here. So returning water into oxygen, and then you're going to be turning ADP to ATP, and then NADP to NADPH. So that is what's going to be happening and what we're going to be discussing in gruelingly specific detail in the next couple of minutes. So light. Well, light is very fascinating because it is a form of energy called electromagnetic radiation. So it's propagated in waves, and the amount of energy is then, of course, inversely proportional to its wavelength. So it can behave as particles called photons, and if you have higher energy, it's going to have sort of shorter wavelengths, and it's going to be blue or violet. If you have lower energy, you're going to have greater wavelengths, and it's going to be red or orange. Now, when a proton actually hits a molecule, there are a couple of things that can happen. The first thing is that it simply can reflect or bounce off, so a reflection. It can go through transmission, which is it passes through that molecule. Finally, there can be absorption, which is adding energy to that molecule. And of course, as mentioned, certain molecules absorb photons at specific wavelengths. The light reactions, of course, are kind of taking all of this right here with the physics into account. So plants can only absorb specific wavelengths of light, and we're talking about visible light over here. And plants are mostly green because they don't absorb green, so they absorb best, according to this particular spectrum over here, around the sort of reddish range and the purplish range. So if we're looking at absorbance activity, it's highest around the purple-blue range and the red range, and not so much in the green range. So the absorption spectrum of chlorophyll, which is shown here in the green, is actually much smaller than the action spectrum of photosynthesis, which is this blue line over here. And this suggests that there are other light-absorbing molecules aside from chlorophyll. But we will focus mostly on chlorophyll in the scope of this course. So here's just a figure that shows you your chloroplast, and you're going to have light reactions that are occurring here on the thylakoid and parts of this over here, and then we have light-independent reactions that are going to be occurring in the stroma. Now, chlorophyll is going to be embedded in the photosystem that's actually on this membrane on the thylakoid, and it's quite similar to the heme group in hemoglobin. But let's go through the entirety of the photosystems and the electron transport chain so we can get a basic understanding of what is going on in this part of the thylakoid membrane. So we're talking about energy transfer here. The great question is, of course, how does photosynthesis convert light energy into chemical energy? What exactly happens? Well, we're looking at energy transfer and that electron transport. So think back to the electron transport chain in the mitochondria. Something similar happens in the chloroplast. At the reaction center, the light energy is going to be converted into chemical energy. And these chlorophyll molecules, which are hydrophobic, are going to be buried within the photosystem. So at the reaction center, we have some reducing agents and oxidizing agents. So think back to your redox reaction lecture. And we have excited chlorophyll A molecule, which is designated by such, as well as the electron accepting molecule, which is designated by this letter A over here. So the first thing that occurs in that whole series is the photosystem, more specifically photosystem 2. So photosystem 2 comes before photosystem 1 in terms of electron acceptance. So we have this chlorophyll plus molecule which loves electrons, and it actually loves electrons more than water. So what it does is it actually strips electrons from water, releasing oxygen as a byproduct. The electrons from water is then going to be taken up and passed into this A molecule. This A minus molecule, so it's passed from it's taken from water, which forms oxygen. Those electrons are going to be passed to an A minus molecule up here. That A minus molecule hates electrons, so it's going to pass the electrons, right? It's going to donate the electron to the electron transport chain. So it's going to be passing from this photosystem 2, and it's going to kick them into the electron transport chain. So donating electron to electron transport in the photosystem 2, and then bumping it into the electron transport chain, which is just over there. So here you go. You have that electron that's being spun up in the energy chain, and then it's going to be passed to the electron transport over here. So afterwards, the chlorophyll returns to the ground state, and we have the 2 electrons that are taken from H2O and oxidized, and we ultimately form these molecules over here. So we have these reactions that are occurring in this photosystem. So this is just a repeat of what I just mentioned. Now the electrons are going to be sent to the electron transport chain, which is over here, and here it's going to allow for the creation of ATP. So the net of photosystem 2 is going to be ATP, which is shown over here, as well as oxygen. And those electrons are going to be then bumped into photosystem 1. So we start off with photosystem 2, and then we go through the electron transport, and then we bump it into photosystem 1. Okay, so we get past, the electrons get passed into photosystem 1. The electrons in photosystem 1 are going to be spun up in energy again, and they're going to be transferred to another sort of electron transport chain type deal, where we are going to eventually make NADPH. So the net of photosystem 1 is going to be NADPH. So if we look back, the net of photosystem 1 and photosystem 2, again, photosystem 2 happens before photosystem 1, but photosystem 2 and photosystem 1, the net gain is ATP, oxygen, and NADPH. So that's how we should get things started. Now, one thing that we should mention again is that all of this is occurring in the thylakoid membrane. But there's also another component to it, which is the acidic compartmentalization, and there are going to be hydrogen ions and light. So one of the things that we have to discuss is that in order for this energy transfer to occur at the very beginning, right, we actually need light. So we need that photon in order to get this whole process started. So it needs light to get started in photosystem 2, and therefore pass that electron from photosystem 2 to the electron transport chain to photosystem 1, and then eventually to another electron transport that makes NADPH. So in order to get the entire thing started back in photosystem 2, we need that light, that photon of light, in addition to water, obviously. And you can see that's how it is according to the photosynthesis equation. So that's great and all, and we have this particular aspect over here in the thylakoid membrane. But what about this compartment? What about this this hydrogen ion component? Where does that come in? Well, if we look at the whole system, this is the components of the electron transport chain, and we can see that the thylakoid interior over here has a high concentration of hydrogen ions, whereas out here in the stroma, there are low concentrations of hydrogen ions. So we have this gradient that's basically maintained, and by stripping water of hydrogen ions, right, we release oxygen, but some of those hydrogen ions get pushed into the thylakoid interior. And we also have hydrogen ion transfer in this particular section over here. And I think this is actually supposed to be over here. I think that they made a mistake with this particular chart. But either way, we have hydrogen ions that are going to be pumped through as well. And so we have an increase, a high concentration of hydrogen ions in the interior of the thylakoid. That makes it a little bit more acidic, but in terms of the concentration, we're maintaining a gradient. So the hydrogen ions are then going to be flowing down the gradient through an ATP synthase over here. Think back to ATP synthase. And this ATP synthase is going to be making ATP. If we look at the entirety of this particular electron transport, though, we can see there are a couple of components. We have plastiquinone over here. We also have, first of all, we have our photosystem too, and then it's transferred to plastiquinone. It's then going to be put into a cytochrome B6F complex, so CYT. Then it's going to go to plastocyanin, which is going to be pushed into photosystem 1, and then to ferredoxin, and then NADP plus reductase, which is what ultimately makes NADPH. So again, looking at the grand scheme of the pathway here, we're taking water and light, right, and we're stripping those electrons from water, and we're releasing O2 and those hydrogen ions in the process. And again, we need light to mediate this process to get started with the help of chlorophyll. Those electrons are going to be starting in photosystem 2. They're going to be passed on to this particular purplish protein over here, this carrier protein called plastiquinone, which I guess is very similar to ubiquinone in the mitochondria. So it's passed to plastiquinone. It's going to be passed then eventually from plastiquinone to this complex over here, the CYT complex, which is called cytochrome B6F complex over here. And then the electrons are going to be passed on to a plastocyanin over here. And then it's going to be passed on to photosystem 1 over here. And then it's going to be passed on to ferredoxin over here. And finally to NADP plus reductase, which eventually helps make NADPH. In addition, by maintaining this gradient, we can allow for hydrogen ion flow down the gradient from the interior of the thylakoid to the exterior into the stroma, and this coupling of the ion flow, of hydrogen ion flow from the interior of the thylakoid to the stroma allows for creation of ATP from ADP and inorganic phosphates. So ultimately, this pathway induces ion flow and allows ATP synthase to make our very much needed ATP in the plant. Now, this pathway induces ion flow through the ATP synthase to make ATP. That's pretty straightforward, but ATP itself is formed here by photophosphorylation, which is a chemiosmotic mechanism, which should make sense. And H plus is transported across the thylakoid membrane into the lumen, creating an electrochemical gradient. So that's pretty straightforward, and it should make a good amount of sense to you. So here's the grand scheme of it and how it's working. So we have the thylakoid, we have the interior, and we have the stroma here. Again, this electron transport process is kind of similar in many ways to that we saw in the mitochondria. Of course, one thing you might want to keep in mind is that in the mitochondrial matrix actually has lower concentrations of hydrogen ions, whereas in the chloroplast, the thylakoid interior actually has a higher concentration of hydrogen ions. So the placement of the ATP synthase might appear to be inverted, if that makes sense. So you should look at this as well as look at the mitochondrial electron transport chain and be able to see what differences there are between the two of them. So there you go. That's the grand scheme. Now, if we look more into the synthesis of carbohydrates, one of the big questions, of course, is what exactly happens? So we have carbon dioxide fixation, which is basically converting that carbon dioxide to a more oxidized form to a reduced form. So carbon dioxide is very much oxidized. We're going to be converting it to a reduced form that's now going to be very much organic. For instance, carbohydrates. And carbohydrates, these sugars, can be used by biological organisms. And then, of course, those carbohydrates are very, very useful for whatever the plant is going to use them for, as well as for whatever the heterotroph is going to eat that plant and use that for. So that's very important. Of course, carbon dioxide fixation kind of starts in light-dependent reactions, but ultimately the synthesis of sugars itself is going to be occurring in the light-independent reactions over here in the Calvin cycle. So that's basically the light reactions, and we're going to talk a little bit about the light-independent reactions, or the dark reactions, aka the Calvin cycle, in another video. So there you go. I hope you found this useful, and best of luck studying, and I will see you in the next video.